Hello student, good afternoon. We will discuss on the control points of the gluconeogenesis. 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 Control of gluconeogenesis. Gluconeogenesis is control at the first step of pathway. First step of pathway. The carboxylation of pyruvate to oxaloacetate. The enzymes catalyze these reactions. Pyruvate carboxylase is activated by acetyl coenzyme and inhibited by the ADP, adenosine diphosphate. And excess of acetyl coenzyme occurs only if the Krebs cycle is proceeding at a maximal rate. Pyruvate is carboxylated to oxaloacetate rather than decarboxylated to produce more acetyl coenzymes. The oxaloacetate is converted into glucose, which either is stored as glycogen or is released to the circulation used by other tissues under the conditions of high yields of ADP, adenosine diphosphate, which indicates that the cell needs energy. Cell needs energy in the form of ATP. Pyruvate is converted into acetyl coenzymes for oxidation by the Krebs cycle. The second control point of gluconeogenesis is the reaction catalyzed by functions 1,6 bisphosphatase. This enzyme is activated by ATP and inhibited by AMP and fructose to 2,6 biphosphate. Fructose 2,6 biphosphate increases the Km of the enzyme for fructose 1,6-diphosphate, thus decreasing the reaction rate. There is also control at the of phosphorylation, dephosphorylation of the enzyme fructose 2,6 bisphosphates as shown in figure. Note that the controls of fructose 1,6 biphosphate is exactly the opposite of that for the glycolysis enzymes fructokinase. Gluconeogenesis is controlled also at the transcriptional level by a number of hormones. In liver, insulin decreases the rate of transcription of the genes coding for the 4 enzymes unique to this pathway: pyruvate carboxylase, phosphoenol pyruvate carboxylate, fructose 1,6 bisphosphatase, and glucose 6 phosphatase. Cortisol, epinephrine, glucagon, starvation, and diabetes have the opposite effect on the transcription of these genes. Although gluconeogenesis takes place in kidney, gluconeogenesis takes place in kidney. Neither insulin nor glucagon affects the renal enzymes level. Affects the renal enzymes level. Of the four enzymes, the concentration by pyruvate carboxylase is the least affected by the changing hormonal and nutritional states. This is because the conversion of pyruvate to oxaloacetate is required for fatty acids biosynthesis and replenishment of the Krebs cycle, as well as gluconeogenesis of the gluconeogenesis. This is the control points of the gluconeogenesis. And regulation of carbohydrates metabolism. We have to study the regulation of carbohydrates metabolism in this period, and oxidation unsaturated fatty acid, control points of gluconeogenesis. This is the 3 points we have to study in this lecture. Okay, thank you.